Kristen Drescher. I'm at Creighton University in Omaha, Nebraska in the United States. And I'm here to talk about creatine and the immune system. And the title of my presentation is "A Creatine Metabolite Alters the Gut Microbiome in a Mouse Model of Multiple Sclerosis." In terms of disclosures, I am a member of the ALSCHEM Scientific Advisory Board, and I do hold a U.S. patent on compositions and methods of treatment of inflammation. Okay. Previous studies from my lab have shown that treatment with creatine or its breakdown product creatinine can influence the sensing and responding phases of the immune system in vitro. And we've done these studies in both mouse macrophages and mouse splenocytes. In terms of the sensing phase of the immune system, we looked at toll-like receptors, which are usually the first indicators that there is some sort of problem or dysfunction in the host. In terms of the responding phase of the immune system, we looked at cytokine production, which are mediators of inflammation and can be mediators of either disease or protection from the host. Okay. In terms of the toll-like receptors, there are 9 different toll-like receptors in mice, and they respond to a variety of different pathogens. They are sensing either things outside the cell, as indicated by these toll-like receptors here, and are on the surface of the cell, or they can be in the endosome. And the endosomal TLRs usually are involved in sensing things like virus infections, so double-stranded RNA, single-stranded RNA, those sorts of things, whereas the external TLRs are looking at things like LPS or bacterial flagellin or lipotylic acid. Regardless of whether they are on the plasma membrane or within the endosome, the TLRs are going to have a sensing, a triggering domain within the cytoplasm of the cell. And previously, I'm just going to give you a very brief overview of what we've done in the past. We compared the response to various types of creatine formulations in terms of how much TLR expression there is relative to control levels. So we used, in this experiment, creatine ethyl ester, which is allegedly a very stable component of form of creatine, kind of your standard creatine, and then a breakdown product of creatine, which is creatinine. And what you see is over time in vitro, when most splenocytes are exposed to either creatine ethyl ester, creatine, or creatinine, is that when you expose either these cells to either creatine or creatinine, you have a downregulation of the toll-like receptors. And that would suggest that they are less efficient at sensing and inducing an inflammatory response. And then just to draw your attention to whether that those mRNA levels that I showed you on the previous slide are translate to protein levels. This is immunofluorescence staining. And what you see here is kind of normal levels, untreated cells, and then creatinine treated cells on this side of the panel is a downregulation or a reduced level of protein expression in the cells that are creatinine treated. Okay. In terms of the responding portion of the immune system, we looked at TNF alpha levels. TNF stands for tumor necrosis factor alpha. It's a very potent pro-inflammatory mediator. It's one of the first mediators that is upregulated when there is an inflammatory response. It's considered to be an acute phase reactant. So TNF, IL-1 beta, and IL-6 are all produced in the liver, and usually within minutes of a threat to the host, these are upregulated. They are all responsible for the fever response that we see in the host. And it's a very important mediator in triggering the adaptive immune response, because it can upregulate surface molecules on immune cells that are going to be involved in getting a very specific response by the host, an antigen-specific response by the host. So it's important both in the acute and the chronic inflammatory response. Okay, and in this experiment here, we looked, this is an ELISA, and what we found was we treated, so we had control cells, and these are mouse macrophages, and they have a baseline level of TNF that's produced. And when you expose them to LPS, which is a very, very potent inducer of inflammation in almost every cell type, what you see is a very high level of LPS, of TNF that's expressed relative to the control. When you just expose the cells to creatinine, which is a breakdown product of creatine, you see a very low level of expression of TNF alpha. But when you take those same cells and expose them to LPS and then creatinine, what you see is that creatinine downregulates the pro-inflammatory response that's induced by LPS very significantly. And the p-value of that is 0.001. So this indicates that creatinine has the ability to reduce ongoing inflammation. Okay. So these were the basis of our studies that we started out with. And this is just immunostaining to show you basically the same thing. When you have cells that are exposed to LPS and creatinine, you see some degree of staining. LPS alone, very high levels of positive stainings, whereas when you have creatinine alone, in the absence of LPS, very minimal staining for TNF alpha, and the D panel is just your isotype control. And this is looking at staining intensity, doing imaging on those cells, and what you see again, LPS induces very high levels of TNF, and when you add creatinine to those LPS treated cells, you have a significant downregulation in the level of TNF alpha staining. So how we propose this mechanism of action is we have a situation where under normal conditions, you have your TLRs, you have your sensing domain out here in the green, and then you have your signaling domain here that's going to be in the cytoplasm. When you treat those cells with either creatine or creatinine, you have a downregulation of those TLRs. So you have fewer TLRs on the surface of the cell or in the endosome. And the signaling pathway that TLRs take is normally going to be the NF-kappa B pathway. And the NF-kappa B pathway, when you have triggering of the signaling domain, you have interaction with I-kappa kinase that in normal situations will take your inactive form of, and your cytoplasmic form of NF-kappa B and degrade I-kappa beta, which is going to keep the subunits of NF-kappa B in the cytoplasm. So when that's degraded, that permits the NF-kappa B to move into the nucleus and basically bind to the promoter of pro-inflammatory mediators, indicating that you're going to get production of things like TNF alpha, IL-6, and other immune mediators. So we did an ELISA taking nuclear extracts of NF-kappa B from the nucleus of cells that were exposed to either creatine or creatinine. And what we found was there was a decrease in the import of NF-kappa B into the nucleus of cells exposed to either creatine or creatinine. In terms of what is the role of TLRs in autoimmunity, there is quite a bit of evidence that suggests that overstimulation of the toll-like receptors, which are going to lead to the production of pro-inflammatory mediators, are going to be implicated in certain autoimmune diseases. And some of those autoimmune diseases are listed here: rheumatoid arthritis, multiple sclerosis, systemic lupus, are all involved with overstimulation of the TLRs. So what we wanted, the outstanding question that we had based on our previous studies is, can the in vitro findings be recapitulated in vivo? Okay, and since my past life was as an involved in MS, we chose to study this in the context of a mouse model of multiple sclerosis. And there is a mouse model of multiple sclerosis that's induced by Theiler's murine encephalomyelitis virus, or TMEV. And this serves as a model of primary progressive multiple sclerosis. So multiple sclerosis is going to be a disease that causes significant alterations in the ability of axons to transmit impulses. So it's going to be a motor disease. So people have defects in, things like walking and movement and things like that. So TMEV is a coronavirus. It's a positive stranded RNA virus. So it belongs to the same family as viruses such as the Group B Coxsackie viruses and polioviruses, which you're probably more familiar with. And when you inject TMEV into a mouse, it induces an acute encephalitis in all strains of mice. So that happens between days 3 and 10 post-injection. And this acute encephalitis is caused by rapid replication of the virus in neurons, in particular in the hippocampus. Later in the disease, it can induce a demyelination and inflammation of the spinal cords in susceptible strains of mice. And mice are susceptible depending on the class 1 MHC molecules that are expressed by the mice, in particular, the H2D region of the class 1 MHC. And you start to see demyelination and inflammation in these mice at day 21 post-infection. And this is induced by the virus. And then as time goes on, you have an autoimmune reaction that's referred to as epitope spreading that is more autoimmune in nature because the virus starts to kind of peter out in the spinal cords of the animals. So alteration, what we know about this model are a lot of things. It's very well characterized. You can knock out various genes. You can supplement with various recombinant cytokines. And what you can find is that alterations in the immune system can impact the level of inflammation and demyelination in these animals. Okay. So things we were considering when we were moving to an animal model was that pro-inflammatory mediators exacerbate disease in mice susceptible to spinal cord inflammation and demyelination. So if you take a susceptible strain of mouse and you give them, for example, anti-TNF, you have much less inflammation and demyelination in the animals. If you take a mouse that's resistant to the spinal cord pathology and you take TNF out of the equation, what you find is that you are changing the level of pathology. So they are more likely to be susceptible to pathology than an animal that is not devoid of TNF. So there's a balance between the various components of the immune system. And the other thing that we were very cognizant of was that when you take creatine and you put it in an aqueous environment, it's going to break down into creatinine. So these were all things that were involved in how we set up the experiment. So our hypothesis was that TMEV infected mice treated with creatinine will have less CNS inflammation than control treated infected mice due to alterations in the gut microbiome of the mice. And why we were interested in the gut microbiome was the fact that a lot is known about kind of globally the immune system, but we wanted to do something novel in this particular study. Okay, so when you think about the immune system and the microbiome, there are a number of things to think about. And the first is when you have a person who is well or an animal is well, it's a symbiotic relation between the microbiome and the host, and you have really limited inflammation. If you do things to interfere with the microbiome, so very commonly one thing that's done is infections, exposure to antibiotics, that sorts of things, you have a situation where you can end up with a host with low-grade inflammation, okay? And then when you have something that's called dysbiosis, what you have is if you interrupt the normal symbiotic bacteria, other bacteria will take their place, okay? So they'll fill that niche. And you can end up with a situation where you have chronic inflammation, okay? So we know that the microbiome has interactions with the immune system. It's been implicated in conditions such as irritable bowel syndrome, metabolic syndrome, so individuals who have obesity, and certain neurodegenerative disorders. If you take a germ-free animal, they oftentimes have immune dysfunction. So how does the gut microbiota impact autoimmune disease? So in this figure here, you have the gut lumen, and what you have is commensal antigens, okay? And those commensal antigens can influence the area outside of the gut or within the gut and interact with cells called T regulatory cells. And T regulatory cells are important because what T regulatory cells do is they shut down autoimmune disease. So individuals who have a defect in T regulatory cells oftentimes have autoimmune disease syndromes, okay? And they're very serious, and oftentimes the patient will die very, in the first 5 years of life or so. So these T regulatory cells produce cytokines such as TGF-beta, transforming growth factor beta, or interleukin-10. And what these do is they tend to modulate the production of cells, in particular, Th17 cells and Th1 cells. So these are CD4 positive T cells or helper T cells, but what they are very pro-inflammatory in nature. So in the absence of these T regulatory cells being primed, being exposed to their cognate antigen, you have a situation where bad things happen to the host, okay? When you have a situation where you have an impaired gut barrier, you have production of cells that are triggering antigen-presenting cells to prime naive T cells, and that results in an increase of Th1 cells and Th17 cells. So leaky gut is going to potentially impact the production of disease in a highly negative manner for the host. Okay. More recently, we know that the microbiome has interactions between the gut and the brain. The brain is an immune privileged site. There's something called the blood-brain barrier, where there's very tight junctions that are formed by resident cells in the brain, and it prevents lymphocytes from trafficking into the brain. So why do you want that to happen? Well, most of the cells in the brain are not going to ever be replaced. They don't go into cell cycle. They're terminally differentiated. So if you lose a neuron, you lose an oligodendrocyte, which is responsible for production of the myelin, it's gone forever in most cases. Okay, it's hard to get an oligodendrocyte to go back into cycle. Okay, so it's a way to protect the host. Okay, if you disrupt the blood-brain barrier, it's going to permit entry of lymphocytes in the brain. And that includes your Th1 cells and your Th17 cells that are highly pro-inflammatory, that produce mediators that can directly damage cells in the brain. Okay. And there have been studies in humans where they've noted that intestinal barrier dysregulation has been associated with certain neurodegenerative diseases, including Parkinson's disease, Alzheimer's disease, and multiple sclerosis. Okay, so it is, this is sort of the basis of where we're going in this study. Okay. So the hypothesis is that where we're at right now is that the creatine breakdown product, creatinine, promotes anti-inflammatory effects on the microbiome of mice with TMEV-induced demyelinating and inflammatory disease. Okay, so our experimental design was that we took creatinine, and we used creatinine because we know that creatine breaks down very quickly in water. And we fed that to mice starting at day -7. So before we injected them with TMEV, and that was prepared in a solution of 20% sucrose. And the sucrose was added just because it's more palatable to the mice. And then our control mice got water with 20% sucrose. So then on day 0, so after a week of feeding the mice those solutions, we injected them with 2 × 10⁵ plaque-forming units of TMEV intracerebrally. So you just inject it into the brain. The virus has a natural affinity to go to the neurons when you do that, in particular in the hippocampus. And then we collected tissue at 2 points. We took the brain at day 7 post-infection, and as if you recall, that is during the acute phase of infection, where you have the encephalitis disease. And then on day 35 post-injection, we took the spinal cord, the brain, and we also collected feces from the animals. And so on day 35, that's when you start to see that's about a week and a half after you start seeing inflammation and demyelination. Okay, so the water was provided ad lib to the animals, and we measured water consumption 2 times a week. Both groups of mice ingested similar amounts of water. The interesting thing about this whole experiment was that the control and the creatinine mice ingested significantly more water than expected, resulting in a dose of creatinine significantly higher than intended. So they were getting about 50 mg per kg of creatinine. And when we originally dissolved the creatinine in the water, we had consulted, in addition to what was published, we talked to several suppliers of mice, and they said, probably about, 3 mils of water per day per mouse would be ingested, but apparently mice have a very, very sweet tooth, and they really enjoyed the sucrose in the water. So they took in more water than expected. And so this is what's happening at day 7 in the brains of mice in the control and the creatinine fed mice. So normally, because the brain is an immune privileged site, you don't see lymphocytes in, you don't see meningeal inflammation. And what you see in this panel here is that the sucrose-fed mice have quite a bit of lymphocyte infiltration in the meningeal surface of the brain, whereas that appears to be way decreased in the brains of mice that got sucrose and creatinine in their water. When you move on to day 35 post-infection, we did CD3 staining for in the spinal cords, and CD3 is what's referred to as a pan T cell marker. So it's going to pick up all T cells, so CD4 positive T cells and CD8 positive T cells. And what you see here in the sucrose control animals is you have quite a bit of T cells staining positive. What you're looking for here is what we call ring staining. CD3 is on the surface of the cells. What you can see is the T cells are outlined, indicating that it's positive staining. You don't just see the meningeal surface, but you also see within the spinal cord, the white matter of the spinal cord, which is going to be where the myelin is, a number of T cells that are infiltrating that. When you go over to the sucrose and the creatinine treated animals, you see much less staining. Most of the staining that you observe is on the meningeal surface, as indicated by the red arrow, and you don't see cells that are infiltrating into the spinal cord white matter, indicating that there would be less dysfunction of that spinal cord. Okay. So the histology findings are indicated here. TMEV infected creatinine-fed mice had reduced levels of meningeal inflammation at day 7 post-infection compared to TMEV infected control-treated mice. At day 35 post-infection, decreased infiltration of the spinal cords by CD3 positive T cells was observed in the spinal cords of TMEV infected creatinine-fed mice compared to the control-treated infected mice. Okay, so that was very promising to us. So then we went on to do microbiome analysis because we figured we had a basis to do that. So we took those fecal pellets that we collected, and DNA was extracted from them. And then they were sequenced using the Illumina platform, sequencing for 16S ribosomal RNA, which would give us an indication of what bacterial species were in those fecal pellets. And what we were really interested in when we started out were the short chain fatty acid producers, which are probably the best studied group of bacteria that are associated with the microbiome. And so these short chain fatty acid producers are the things that the acids they're producing are acetate, propionate, and butyrate. And in general, butyrate has been probably the most studied of all of these. Butyrate is known to stabilize epithelial tight junction integrity, which is going to be important in ATP production, as well as immune system modulation. And the other interesting thing is about butyrate producers is that if you take butyrate and culture it with biopsies from Crohn's disease patients, so Crohn's disease patients have an inflammation of the intestine, and what they found was that butyrate inhibits NF-kappa B, and that results in a decrease of TNF-alpha, IL-6, and IL-1 beta in a dose-dependent fashion. And all of these cytokines, TNF, IL-6, and IL-1 beta, are pro-inflammatory acute phase reactants. Okay. So what we found when we did the sequencing was we first looked at changes at the phylum level. Okay, and the ones we were most interested in were proteobacteria. And what you see here is that sucrose fed animals had an increase in a significant increase in proteobacteria compared to creatinine fed animals. And proteobacteria is associated with barrier dysfunction. Acintobacteria was the next group that was of interest to us. And what we found here was the creatinine-fed animals had an increased level of Acintobacter compared to the sucrose-fed animals. And this phylum is associated with maintaining and repairing barrier integrity in the intestine. The last group that we were really interested in was the butyrate producers, and this is represented by the Firmicutes. And what we found was no significant difference between the creatinine and the sucrose-fed animals. But the reason we were interested in the butyrate producers is because of their role in barrier integrity and anti-inflammatory molecules. So we then looked at the genus level, and this is looking at the phylum Proteobacteria, which is associated with barrier dysfunction. Okay. What we found when we looked at the genus level were two significant changes, and those were Parasuterella, which is shown here, sucrose in the orange, and creatinine in the blue. And what we saw was a significant decrease in the level of Parasuterella in the creatinine-fed animals. Parasuterella is pro-inflammatory and associated with chronic inflammation. The second group that we looked at was the Escherichia Shigella group, which includes E. coli, which I'm sure you're all familiar with. This is a pro-inflammatory group of genus, I should say. And what we found was a significant increase in the creatinine-fed animals, but not as big a change as what we saw in the Parasuternella group. Okay. We then looked at the phylum Acintobacteria, and that the genus that had a change in levels of gut population were the Aldricruiza. And what we saw was the sucrose fed animals had a much lower level of this particular genus than did the creatinine fed group. And what's important about this genus is they are known to be anti-inflammatory in nature. They have reduced levels of NF-kappa B. Remember that NF-kappa B is going to bind to the promoter of cytokines such as TNF-alpha, IL-6, IL-1, and it's known that this genus reduces IL-6 levels. So anti-inflammatory in nature. Okay. We then looked at the butyrate producers, even though there wasn't a significant change in the Firmicutes phylum. And what we were drawn to in particular were the Lactospiraceae, NK4A136, which was significantly higher in the creatinine-fed group. Other members of that genus were increased in the sucrose group. And the Roseburia is known to be anti-inflammatory, was significantly increased in the creatinine-fed group. It's known to restore barrier function, and it's also known to suppress actually tumor function in the intestine. When we were looking at these various genus of bacteria, we were focusing primarily on bacteria that were known to do something or to influence barrier function. A lot of these bacteria are not well described in terms of how they impact the gut barrier. Okay. So the conclusions that we made were that creatinine reduces pathology in the brain and spinal cords of TMEV-infected mice. Creatinine alters the gut microbiome of TMEV-infected mice. Not all bacteria of a genus associated with intestinal barrier integrity are equally affected by creatinine. Creatinine increases the relative abundance of some, but not all anti-inflammatory bacteria, and decreases some of the pro-inflammatory bacteria. Creatinine decreases the relative abundance of Parasitruella. Creatinine increases the relative abundance of Aldacroizia and some butyrate producers. And because creatinine is a breakdown product of creatine, which is well studied and a safe supplement, we expect that individuals using creatine will experience decreased gut barrier leakage than individuals who do not use creatine. Okay. In terms of future directions, we need to further characterize the microbiome at a species level, because we've gotten down to the genus level, but we need to go one step further to find out exactly which species are being impacted by feeding creatinine. We would like to do a dose response to determine the range of creatinine feeding that can positively impact disease, and do some testing using creatine and creatinine to test the integrity of the intestinal barrier in vivo in a mouse model, as well as in vitro. And ultimately, we would like to utilize germ-free mice that have been reconstituted with specific species of bacteria to understand the specific impact on disease. Okay. This work could not have been done without the help of a lot of people. Corey Leland and Lisa Reisbig. Corey was a technician in my lab who started the in vitro work on TLRs. Lisa Reisbig was a PhD student in my lab, and ultimately she did all of the work looking at cytokine expression and NF-kappa B movement to the nucleus. Brian Wynn was a PhD student in my lab, and he worked with Hannah Flum, who is a very, very talented undergraduate who worked in my lab this past summer. And the two of them did all of the analysis of the abundance of the bacteria in the sequencing that we got back from the Illumina platform. Jennifer Achtung, as well as her husband Thomas, are at the University of Nebraska in Lincoln, and they actually ran the sequencing data for us on the fecal pellets. University of Nebraska Medical Center, Tom McDonald, a brilliant scientist who died recently, is the one who got me into creatine and creatinine, as well as his partner in crime, Steve Tracy, and the conference organizers that do an amazing job. AlsChem, the Dynamic Business and Science Society in particular, Diego Ocampo and Sebastian Sarmiento have been wonderful in helping me get this together. And obviously Rick Kreider and the organizing committee did a wonderful job putting this together. In terms of funding, Hannah was funded by the IMBRE program through the NIH. And I thank you all for listening.